All right, in this lesson we'll take a look at what is known about chromosome separation during mitosis. Here we see a cell, it's an animal cell, and it's in the lineup stage of mitosis. So the chromosomes are in the center, lined up here. They're duplicated already. They were duplicated back in interphase of the cell cycle. And we see a network of proteins that appear to be sort of connected to the chromosomes originating from each side of the cell. The existence of this sort of protein scaffolding suggested one obvious hypothesis for what was going on. Maybe these proteins are pulling the duplicated chromosomes apart. So separation is the result of a pulling force generated by these proteins. Let's take a cartoon look at that cell. It turns out that these proteins are none other than microtubules, the same ones that we studied in amoeba movement. And they originate from the poles of the cell, and there are structures at each side of the cell called centrioles. And so one hypothesis suggested that maybe some kind of force was being generated that would then pull the duplicated chromosomes apart and deliver them to each side of the cell. That suggests that the microtubules were connected to the chromosomes and some pulling force was being generated at either side of the cell. So this was the hypothesis for chromosome separation that existed for years. We could take a look at it this way. In this version, the pulling hypothesis, the microtubules are attached to the chromosomes, and chromosomes will be pulled in opposite directions. But more recently, another possibility emerged with the discovery of a new class of proteins that we'll call molecular motors. Molecular motors are proteins that can carry a cargo and that use microtubules as a track, or like a road. They attach to the microtubules. They can change their shape, kind of like an inchworm inching its way down a branch. The molecular motors are proteins that can move along a microtubule and carry a cargo, possibly a chromosome. So in this view, this new hypothesis, chromosomes were not pulled apart, they were driven apart by molecular motors. The duplicated chromosomes each would be attached to a little molecular motor, like a little engine, and it would inch its way along the microtubule. So the microtubule is a track for this hypothesis. The microtubule, notice, is not directly connected to the chromosome. So we've got rival hypotheses. We've got the pulling hypothesis, and we've got the driving hypothesis. Now remember, these things are small, right? So in this picture, where's the molecular motor? Well, we might not see it, right? So the chromosome, you're just not going to see the molecular motors, so it may look like the microtubule is directly attached to the chromosome, but what you don't see are the little motors, the little engines that are driving them. But nevertheless, we've got now two possibilities here. It could be that pulling is happening. It could be that driving is happening. What we need are some experiments to test these hypotheses. So let's consider one possible experiment. There are drugs that in fact disrupt microtubules. Remember, microtubules are made up of the subunits, the sticky balls that stick together spontaneously, and there are chemicals that interfere with that assembly, right? So one proposal might be, well, let's add a drug to the cell that disrupts microtubules. And let's ask ourselves, would this be a good experiment to figure out whether the chromosomes are pulled or driven? So take a moment to think about whether that is a useful experiment to test the rival hypotheses. Okay, well, maybe you arrived at the view that it is actually a poor experimental design. And the reason for that is because the experiment, that drug that disrupts microtubules, it interferes with both hypothesized mechanisms. Both hypotheses make the same prediction, that the chromosomes will not move, right? Because you can't be pulling the chromosomes without a rope, and the drug would break the rope. And you can't be driving the chromosomes without a track. The drug would disassemble the track. So This is not a good experiment to add a drug that disrupts microtubules, because both hypotheses make the same prediction, that the chromosomes will not move. They won't move under the pulling hypothesis because you can't pull if the rope is chewed up, and the driving hypothesis won't work because you can't drive without a track. What we need is an experiment that blocks only one of the proposed mechanisms. So you might take a moment just to think about some possibilities. Some years ago, some researchers used lasers to do such an experiment. What they did was they used a laser to cut the microtubules on one side of the cell. Right? Remember, we're during the separation stage of mitosis. The laser would cut those microtubules, and the other side provided a nice comparison. Right? So we're interfering with the microtubules here, but we're not destroying the entire set of microtubules. We're simply cutting them. Right? So whereas the drug completely disassembled the microtubules, the laser just cuts them. Now think about what predictions follow from each hypothesis. So remember, our two hypotheses are the driving hypothesis and the pulling hypothesis. So take a moment to think about what each hypothesis would predict. Okay, we can use our reasoning format. If the chromosomes are pulled, and you cut the microtubules halfway, then the chromosomes don't move at all, right? So when you cut the microtubules, you cannot pull if the rope is broken, right? So they should not move at all if pulling is the correct mechanism. On the other hand, if chromosomes are driven and we cut the microtubules halfway, then the chromosomes will move until the cut. Notice, for the driving hypothesis, there is still an intact track here, right? So the motor can drive these chromosomes some distance until the break, and then we might expect the chromosomes to stop. But that's enough of a difference that we can sort of pit these two rival hypotheses against each other. The pulling hypothesis predicts no movement. The driving hypothesis predicts movement until the cut. Here's what that would look like. Again, if you cut if the track is disrupted there, the little molecular motor can't operate, so the chromosome would stop. This is a good experimental design because each hypothesis makes a different prediction, as we've seen. Now, what happened? Turns out the results were that the chromosomes did in fact continue moving. And that supports the driving hypothesis, right? So they moved until the break in the microtubules. But the evidence for driving does not completely rule out the pulling hypothesis. After all, both driving and pulling could be happening simultaneously. To think about that, in other words, it's possible that the chromosomes are being driven along the microtubules, but also the entire set of microtubules is pulling, right, the chromosomes at the same time. For example, when you go to the mall, if you get on the escalator going up, you can just kind of stand there and be carried up, or you can also walk up the escalator. So in that case, you'd be sort of driving yourself and being pulled up at the same time. In the same way, it's possible that chromosomes are being driven and also pulled at the same time. In fact, there is other evidence that pulling is happening as well. So at this stage, then, let's wrap up with a few points here about chromosomes. First of all, they're duplicated prior to cell division. They are organized and moved by the spindle structure, which is the name for all those microtubules that handle the separation of the chromosomes. So they're organized and moved during mitosis, and each daughter cell receives a full set. So at this point in history, then, it was looking pretty good that the chromosomes were in fact important hereditary molecules. The cell went to a lot of trouble to get these chromosomes into their daughter cells. Now, if we think about how the cell is achieving this, it's kind of interesting. the cell is, in a sense, finding chromosomes in a trial and error way. The microtubules, as they start forming during the early stage of mitosis, they in a sense randomly explore the cell cytoplasm. And when they find a chromosome, they are stabilized. And this allows them to find chromosomes anywhere in the cell. Let's see what that would look like. Here we have a little picture here of the centrioles, and we have microtubules growing. Remember, they spontaneously assemble, growing out towards the center of the cell where duplicated chromosomes are located. Now, the idea is, just as we saw with amoeba, the microtubules will be growing and shortening. So they're going to randomly be sort of changing their lengths, getting longer and shorter and so on. But sort of like what we saw in the in the amoeba, the microtubules can be stabilized. And that appears to be happening when they encounter a chromosome. When the microtubules find a chromosome, they are stabilized, and they are going to then be involved in arranging the chromosomes in that lineup stage of mitosis. But this random exploration of the cell is what allows these microtubules to find the chromosomes, even if they're at all kinds of odd locations within the cell. And that is kind of the beauty of this trial and error approach. Eventually, these microtubules, as they sort of metaphorically explore the cell, are going to find the chromosomes. And that will trigger the next lineup stage of the cell cycle, or of the mitosis process. And here we see then the microtubules have already found their duplicated chromosomes, and in fact, the duplicated chromosomes are separating to opposite sides of the cell.